So, first thing to say is we are a small virtual company. The team currently comprises 3 people: myself, the CSO Rebecca Ashfield, David Blackburn, who's a scientific founder, or whose intellectual property was used to develop the technology that we've got. Why Ducentis? The key point for me, the key attraction of working in this field when I first looked at the field, was that it had 3 attributes which I've often worked in programs which are lacking one of the 3 attributes. The first was we had a very novel mechanism of action which we believe will be of interest to pharma. The second is that we had what I would term a tractable drug discovery problem. That's to say, I thought it would be possible to go from the concept to a molecule that would be a molecule that you could administer to patients. And the third point is that clinical need is aligned with commercial opportunity. So I felt that we had ticks in all 3 boxes, and I'd worked in the past on programs where you had 2 out of 3, and that's not a nice place to be. We're currently a preclinical company. We're close to candidate selection. I would say we essentially do have a candidate now, and we're looking to start manufacturing work at the end of this year, the start of 2021. And our exit strategy is licensing or trade sale to pharma. So this is just a brief slide indicating that there is still a great deal of unmet clinical need in the autoimmune space. A lot of existing autoimmune therapies are associated either with loss of activity following administration to patients for 6 to 12 months, or non-responders. So you have, and also in some instances where you're using immunosuppressive agents, you may get toxicity associated with the immunosuppressive agent itself. This is just a brief slide highlighting what the current mechanism of action is of existing marketed biotherapeutic drugs. One way or another, you can put most existing marketed biotherapeutic drugs into one of the 4 boxes I've indicated here. Either ablation therapies such as rituximab, cell trafficking molecules which inhibit cell trafficking, the T-cell co-stimulation molecules, and then finally the cytokines such as Humira. And what we're doing is something we believe to be very novel compared with these existing therapies. Most of the current therapies act one way or another by blocking a pro-stimulatory signal, and what we're doing is fundamentally different to that. So the CD200 axis is an axis comprising a pair of molecules, a receptor that's expressed on the surface of immune cells, and a ligand which is expressed on the surface of cells which, if I can put it like this, need to evade immune responses. So when this CD200 ligand, and our therapeutic agent is the CD200 ligand, or it's a modified form of it, when the ligand engages with the receptor, then you get a deactivating signal, and you get deactivation of myeloid cells and activated T cells and B cells. I've lost my image. So I will just continue talking on the basis that we don't have much time. So what we did when we first looked at this field, because what we're dealing with here is a checkpoint molecule. So what we have is a checkpoint molecule which, at the time that we started we started doing this work, the term checkpoint molecule wasn't in common usage, but that is essentially what we have. So we're flipping the checkpoint story into autoimmune disease. So instead of activating the immune system with a checkpoint molecule, we're deactivating the immune system. And there was quite a bit of pharma interest when I first looked at this molecule in CD200 as a potential therapeutic. You all recovered your sanity? So there was quite a bit of pharma interest in CD200 as a potential therapeutic, but there was no easy route to getting intellectual property on the wild-type molecule. So what we did was to engineer the wild-type molecule using a couple of different approaches. And in essence, we've taken the affinity of the wild-type molecule from about 200, a KD of 200 nanomolar to sub 1 nanomolar, just sub 1 nanomolar. And we've increased the residence time on the receptor from seconds to about 30 minutes. So an increase in affinity and an increase in residence time on the receptor of our engineered molecules. And we filed patents on these molecules, and the patents have secured past the first step, which is to say we've got an international search report of the patent, and the examiner who wrote the international search report finds all of our claims to be novel with respect to the literature. So in terms of proof of concept work, there are 3 areas of proof of concept that we're looking at. The first is GWAS studies, and there are GWAS studies linking CD200 to asthma, multiple sclerosis, and atopic dermatitis. When we started looking at the GWAS studies originally, and I apologize for that, I still don't have the slides, but when we started looking at the GWAS studies originally, they were all merely giving you an association. There was no evidence of causality. And what has now happened is that there's been recently a publication demonstrating causality, in the sense that what you're looking at is coding mutations which increase the incidence significantly of atopic dermatitis in patients with atopic dermatitis. And this GWAS study was then validated by going to the UK Biobank and confirming that patients with atopic dermatitis do indeed have. Okay, so I've moved beyond this slide. So this is just a slide sort of indicating the success of the engineering work that we did. And you can see, reading from top to bottom, that the residence time on the receptors increased and the KD has gone down. So this is then just a slide summarizing what the proof of concept work is that we're doing here. And what we've done is looked at functional and phenotypic readouts on primary human immune cells. We're then also looking at in vivo disease models, and there's a lot of literature data sort of showing that CD200 agonism reduces pathology in a whole variety of in vivo disease models. What we want to do is to show that our molecules are active in these in vivo disease models, and that is problematic because there's no cross-reactivity between the human and the rodent receptor. We're addressing that in a couple of ways. We're developing transgenic mice which express the human receptor, and then we've generated surrogate molecules which surrogate mouse molecules which are high affinity for the mouse receptor, in order to show that when you increase affinity of the molecule for the receptor, that you get more bang for your buck in terms of potency in an in vivo model. And then the translational work, I've just commented on some of the translational work. There is, as I say, very compelling translational evidence in atopic dermatitis and sort of more indicative evidence in RA and asthma. And we're working with two groups of clinicians, with Simon Milling at Glasgow University and Ken Smith at Addenbrooke's, to sort of amplify this data set. Ken Smith in particular has very interesting data with samples from patients with vasculitis, IBD, MS and nephritis. And what we're wanting to do is to demonstrate a link between CD200 expression and pathology in these patients. So this is a summary. I think I've already summarized this, but this is a summary of what we've done with the human immune cells. And in a nutshell, what we do is we reduce monocyte to macrophage differentiation. We inhibit respiratory burst in neutrophils. And in M1 and M2 macrophages, we decrease antigen presentation. We decrease cell trafficking. These are phenotypic responses. These are markers of antigen presentation, markers of cell trafficking, and an indication that we increased Treg activation. And our molecules, the higher potent, our high affinity molecules are all more active than the wild type molecule in cellular assays. And we're differentiated, certainly from Humira, and we're looking at other agents to demonstrate differentiation. So this is a slide which just summarizes where we are in terms of the developability parameters. I'm not going to talk you through that, but I can sort of discuss that with any of you later on. And I think the final slide just summarizes where we are in terms of our funding. So we've done this to date with very modest funding. In 2019, we were the first UK biotech to get funding from the LifeArc seed fund. This was a seed fund that was essentially, I think the seed fund opened in June 18, and we were in sort of late stage due diligence at the end of 18 and secured the funding in June 2019. So we secured 1.5 million from LifeArc, $500,000 from our existing investors, and what we're aiming to at the present moment is to raise a Series A of 12 to 15 million in order to complete clinical trials on the schedule I've indicated here. So we want to do the manufacturing work. We're already talking to CDMOs about the manufacturing. The aim is to finish that in 2021, the GLP tox and regulatory submission in 2022, and then be in patients in 2023. So thanks for your time, and I'm sorry about the loss of the slides.